This protocol is fluorescent in-situ hybridization, or FISH. First, we need to filter seawater onto a filter, and we do this using filter towers. We use a backing filter, it's 0.8 microns, which helps evenness of distribution, and then we use a 0.2 micron polycarbonate filter, and the bacteria stick on top of the polycarbonate filter. Unlike DAPI, we actually don't stain this filter. We just filter straight onto it, and then we take the filter off, and it's mounted onto a slide and stored with desiccant in the -20 freezer until we go to the next step. This filter is then taken and cut into 8 pieces of pie, and each of those little pieces of pie can be probed using a oligonucleotide probe that binds to a specific lineage. So we mount this piece of filter, which is kind of, we call them ice cream cones, using a tough spot onto a filter. We then rinse it in an ethanol wash to get rid of any surfactants on the filter that might impact the actual probing. And finally, we add a small sequence of DNA called an oligonucleotide probe that has a fluorescence signal attached to it. We use Cy3. It manages to get into the cell on a filter and bind to the DNA, provided the complementary strand of DNA is there. So if that lineage is there, this probe will bind to it and it will fluoresce on the microscope. We actually let the binding process happen overnight at 37 degrees in a hybridization oven, and we use a hybridization solution with a specific percentage of formamide that has been predetermined for the specific probe that we're using. And this goes overnight, and then the next morning we actually wash it at a specific predetermined temperature using a wash that's has different concentrations of sodium chloride. The higher the sodium chloride, the more stringent the parameters, or the better binding that you get. For instance, certain lineages are faster growing, and as a result, they can be washed at a higher temperature and with a salt concentration that's lower. We wash for 2 15-minute intervals in a hive wash, and once that's done, we've gotten rid of any non-specific binding that could have occurred. We dry the filter, and then we mount it with a mounting media called DAPI City Floor. So its City Floor can prolong the life of the filter, and the DAPI does the counter staining of the actual bacteria. So when we look at it under the microscope, we can actually see all bacteria using the DAPI stain in UV light. And then because the probe is attached to cyanide, which fluoresces under narrow green light, we actually see the lineage as a red color with a different wavelength. And we switch between the 2 wavelengths, and we take anywhere from 6 to 12 pairs of images. And then with the image analysis, we actually take these images and we do a count of the DAPI and the count of the Cy3, which is the actual lineage, and then we do an overlay to make sure that everything that is lighting up is an actual cell, and we get a percentage. So this lineage is this percent of the total population. And we can do this for quite a number of lineages. So often when we set up for a fish, we're looking at 5 or 6 lineages at a time.